This is a production of Cornell University.
Okay, hello everyone. As you introduced me, my name is Jay. And today I'm going to talk about QTL mapping of potato cyst nematode resistance and two agronomic traits in potato. I performed 3 different research projects evaluating 2 molecular markers for selecting potato cyst nematode resistance clone and QTL analysis of new potato cyst nematode resistance and QTL analysis of chip color and specific gravity. So to help your understanding, I start with introduction of potato cyst nematode. There are 2 potato cyst nematodes. One of them is Globodora rostokensis. Another one is Globodora pallida. They also have a common name called golden nematode and pale cyst nematode, respectively. This picture shows juvenile stage of the golden nematode. And for your convenience for today, I'm going to use these 2 common names for following slide, golden nematode and pale cyst nematode. This pathogen is a soil-borne pathogen, and it has a really stable dormant stage, which is called cyst. According to the one report, the nematode could survive for over 30 years by maintaining this dormant stage. And also it can be easily transferred by farming equipment. So because of these features, once it's infested in a potato field, it is very hard to eradicate or control this pathogen. In the United States, the golden nematode is distributed in New York State here, and pale cyst nematodes distributed in Idaho. Detection of the potato cyst nematode always initiate quarantine action and trading embargo. And this is one of the 3 most serious potato pathogens in the world. According to the APHIS, if golden nematode became widely prevalent without appropriate control, annual loss would reach $4.8 billion. But I think instead of this tax, I think this picture well reflects how scary this pathogen it is. These bags are showing the potato yield from the non-infested field, and these bags represent potato yield from infested field, and you can see like a huge difference. So this pathogen, there's a reason why I started to study golden nematode resistance of the potato. In 1941, the golden nematode was first observed in the United States, especially in Nassau County on Long Island, New York. And after the first detection, golden nematode, especially pest type R1 of the golden nematode, gradually appeared in 8 other counties. At this moment, let me briefly explain how we can divide into the different pest type. For example, if a nematologist takes a sample from the infested field, this is one way actually. There are multiple ways, but the nematologist inoculate the samples on these 3 different wild potato species. And if the nematode can survive on one plant, we can put the plus sign. And if the nematode cannot survive or reproduce, we can put the minus. So by combining these results, we can divide into the multiple pest types. Golden nematode composed of 5 different pest types, and pale cyst nematode is composed of 3 different pest types. And I just want to emphasize that pest type R1 has been predominant pest type for a long time, not only in many European countries, but also in New York State. So many researchers put an effort and time to find resistance genes effective to this pest type. Fortunately, they could find H1 gene, which is very effective against the pest type R1. It is known to be located on the chromosome 5. And the many potato breeding programs in the world using these genes was really successful. For example, Cornell breeding program solely could release 24 potato varieties, which include the H1 gene. And of course, these potato varieties works well to control the damage of the pest type R1. However, there was one drawback in the Cornell potato breeding program when it comes to the R1 resistance, because we still relied on time-consuming bioassay. So to reduce the amount of the bioassay, maybe marker assist selection is one of the good options. Do you know that? So I start to evaluate these two markers, which known to be tightly linked to the H1 gene. So from now on, I'm going to explain how I evaluate the two molecular markers for selecting R1 resistance potato clones. This picture shows PCR result of 57R marker and TG689 markers. Pike, this is one of the cultivar of the potato, and this is known to be resistant against the R1, and Catadin is susceptible cultivar. So I use these two cultivar as both resistance control and susceptible control. As you can see in this picture, resistance clone produce 450 base pair PCR product, and susceptible clone produce 1,500 base pair PCR product. And when I tested other breeding clones or cultivars, I could get the same result like this way. When I tested TG689 marker from the resistance clone, I could see 114 base pair PCR product, and I could not see any PCR product from the susceptible clones. Because of that, I also use BCH marker as a PCR control to check the PCR quality. So for your convenience, I'm going to use plus sign for presence of the 400 base pair when it comes to the 57R PCR test. And I'm going to use negative sign for presence of the 1,500 PCR product when I'm talking about the 57R marker. And I'm going to use plus sign for presence of the 14 base pair PCR product when I talk about TG689 PCR test, and absence of this PCR product will be marked as a negative sign. So I first tested 38 potato cultivars. And as I expected before, the markers work quite well. For example, when I tested all 20 susceptible cultivars, all PCR tests show negative result. And when I tested 18 resistance cultivars, 17 of them shows positive for the 57R, and 14 clones was positive for TG689. Of course, there are some exceptions such as Andover, Eva, and King Harry. Andover shows negative sign even though this is R1 resistance, and Eva and King Harry show the negative response from the TG689 PCR test even though those are R1 resistance. So I suppose that this caused by the recombination event. So now I start to talk about how I tested cornell breeding clones. My purpose was to test whether the two markers work well in a cornell potato breeding program or not. So I collected 342 cornell breeding clones and performed the exactly same test, bioassay and 2 PCR tests. And this table summarizes all the results. As you can see here, there are multiple combinations. So depending on the combination of the result of the PCR test and the bioassay, I could divide into the 8 different cases. For example, if a clone was resistant in a bioassay and showed a positive result in a 57R and the TG689, the clone belonged to case 1. And if another clone, which is susceptible and showed a negative response from the 2 PCR tests, the clone belonged to case 8. So as I expected before, majority resistance clone belong to case 1, and majority susceptible clones belong to case 8. And rest of them caused, I believe, because of the recombination. So I'm going to call from case 2 to 7 as a mismatches for your convenience. So majority of mismatches shows a relatively small number, 8, 6, 1, or 0, and so on. But when I saw the case 4 compared to the other mismatches, case 4 shows relatively higher number. So I'm going to come back case 4 in the following slide. Once I test the 342 clones, I also calculated prediction, positive prediction value and negative prediction value. Positive prediction values are calculated by determining the percentage of the clone with the marker that were resistance. Negative prediction values were calculated by determining the percentage of the potato clones lacking the marker that were susceptible. So as you can see in this table, both markers really well, especially for positive prediction. This means once you saw the positive response from the PCR test, the clones has a really high chance to have H1 gene in it. On the other hand, negative prediction was much lower. However, this cannot be a big problem for the breeding purpose because potato breeders just focusing on selecting resistance clone. We are not interested in selecting susceptible clones. So we conclude that from this result, the two PCR marker 57 or TG689 can be applied into the future Cornell breeding program. Okay, now let me go back to case 4 again. So as you can see here, even though these clones are resistance, they show negative response from the two PCR. So to figure out what happened here, I go back to the list of the case 4. So second column shows the list of the case 4 clones. And third column and fourth column shows parents' information of each clone. And while analyzing this table, I realized that the clone E48-2 frequently appeared as either male parent or female parent for these clones. So I reperformed the same bioassay and the two PCR tests for E48-2. Interestingly, I could not see any difference compared to the general R1 resistance clone. So this shows resistance in a bioassay and also plus response from 57R and then TG689. So quite similar to the general R1 resistance clones. So I go back to the pedigree information of this clone. And interestingly, I realized that Solanum virtuality, this is one of the wild potato species. And these features predominantly in the pedigree of the E48-2. According to the Dalamo et al., some accession of these wild potato species are resistant to R1. So I suppose that previous breeders introgressed an unknown R1 resistant genes came from the S fertility. So I thought that E48-2 include not only H1 gene in it, but also another resistant genes located on another chromosome maybe. So it results in those kinds of mismatching. So this is all for first research project. So now I'm going to explain QTL analysis of new potato cyst nematode. New potato cyst nematode resistance. As I mentioned before, pest type R1 have been predominant pest type for a long time in New York State. However, in 1995, the new pest type R2 was first discovered in the Steuben County in the New York. And the problem was that the H1 resistance is not effective against R2. There's a reason why I started QTL analysis of R2 resistance. For this analysis, for this research project, I prepared the Y36 population, which came from the cross between NI121 and NI115. These two parents are auto tetraploid potatoes, and both of them are heterozygous potato. And NI121 is R1 resistance, and NI115 is R2 susceptible. For QTL mapping, I use 182 progeny. For genotyping, I use a SourceCap SNP chip. This is quite useful too because this is composed of 8,303 SNP position. So I can have a higher chance to find some QTL position. Also, reference for each SNPs are available from the SPOOD database. So you can find the location of the SNP position such as chromosome number, physical map position, and also short description for related genes. So after scanning the DNA sample of the Y36 population with this SourceCap chip, I could get the raw data, which is called theta score or theta value. Theta score is like a number, so I have to transform from theta score to general genotype data, such as AAAA or AAAB, AABB, and so on. So after transforming the genotype data, I load on one software which is called tetraploid SNP map. Tetraploid SNP map is specially designed for QTL analysis of autotetraploid. So after loading the selected SNP marker on this software, as we can imagine, all markers are mixed each other, and through the clustering function belong to this software, I could collect the SNP markers which are on the same chromosome. So potato composed of 12 chromosomes. So each circle represents each chromosome in this case. So through the ordering step, I could rearrange the selected marker which are on the same chromosome in an order like this way. And then through the phasing step, I could decide which of the 4 homologs each marker resides on, like this way. At this point, unlike the diploid QTL analysis, like a general diploid QTL analysis, some manual processes are necessary for phasing step in the autotetraploid QTL analysis. So compared to the diploid QTL analysis, this process takes more time. So after completing phasing step, I could get linkage map. This is one of the example of the linkage map. Since I crossed the 2 heterozygous parents, I could draw 2 different sets of the genetic map. In this case, homolog 1 to 4 represent 4 homologs of the NI121, and homolog 5 to 8 represent 4 homologs of the NI115. And left side representing the genetic map, all SNPs markers are combined. At this point, I'm going to share the challenges that I encountered while performing the autotetraploid QTL mapping. Unlike the general diploid SNP calling, tetraploid SNP calling a little bit difficult. Let me share one example. In this case, the graph showing the theta score value. So x-axis represent theta score from 0 to 1. And you don't need to worry about y-axis in this case because these y-axis relate you to the quality issue of the SNP marker. So if a clone has an AAAA genotype, generally the clone centered on the 0. And if another clone has a AAAB, that clone's generally centered on 0.25. And if another clone has a AABB, the clone's centered on 0.5 and so on. So if the clones are well distributed, like the first example, we can easily assign appropriate genotype for each clone. However, look at this example. As you can see here, even though there are multiple clusters, it's not easy to assign appropriate genotype. So I spent a lot of time for like a best or the appropriate assigning best genotype for each clone. So I have been struggling for a long time. But fortunately, in 2017, Jeffrey Endelman released a new R package called the cluster call, and I can overcome this difficulty. Also, before the tetraploid SNP that I used for this QT analysis, there was only one software which is called tetraploid map. Actually, this is previous version of the tetraploid SNP map, and it has a lot of limitations. One example would be the number of the SNP marker that I can use. Only less than 50 SNP markers can be used for each chromosome if I rely on this software. That means I can lose many chances to find significant QTL peak. So before that, I could not find significant QTL peak. But once I started to use tetraploid SNP map, the US version released in 2017, I could find significant QTL peaks related to the R2 resistance. And finally, there are still manual steps during the linkage mapping. So as I mentioned before, compared to the general diploid QTL mapping, autotetraploid QTL mapping takes more time. So thanks to the newly released softwares, I could get the excellent genome coverage. In my case, I could select 2,761 SNP markers to cover 12 chromosomes. And as you can see here, several hundred markers can cover each chromosome. That means I can have a higher chance to find QTL peak. And look at the map coverage. As you can see here, majority of them are well covered by the SNP markers, around like a 99%. So as a result, I could get really excellent genome coverage. This picture shows a SNP marker that I use. The green color represents SNP marker segregated in a NI115, and red color represents the SNP marker segregated on a NI121, and black color represents SNP marker segregated in both parents. And as you can see here, SNP markers well distributed across the chromosome. Okay, now to complete the QTL mapping, I had to load the phenotype data on the tetraploid SNP map. So I'm going to explain how I measure the potential cyst nematode resistance. After I prepared the Y36 population, I planted into each pot in a greenhouse. And then after 2 weeks later, I inoculate 8,000 eggs into each pot like this way, and then kept growing for 10 weeks. Once the plants start to wilt, I transfer all soil and plant material into paper bag and have wait for 1 month for drying period. Once all samples are completely dried, the samples are transferred to the Fenwick funnel. This silver color represents the Fenwick funnel, specially designed for separating cysts from the dirt. So with the high pressure water and this funnel, I could separate cysts from the dirt and plant materials. The collected cysts were counted under the microscope like this way. So as I mentioned before, this biopsy takes a lot of time. For example, I had to spend 3 months for growing and inoculation, and another 1 month for drying period. And based on one labor force, Fenwick funnel process can be performed only for 20 to 25 samples per day. And counting process can be performed less than 20 samples per day. So in my case, I used a full replication. So I had to grow around like 860 samples in the greenhouse. So as a result, to complete this whole process, I had to use around like 1 year. So as you can see here, reducing this bioassay amount is really important for selecting nematode resistance clone. After getting the original phenotype data, I transformed to the log 10 because original phenotype data skewed toward the right side too much. Once I transformed, I could get the distribution close to the normal distribution. And I load this data on the tetraploid SNP map. The two pairs located here and here. And this is one of the examples of the QTL map, especially chromosome 5 QTL map. And in this case, x-axis represent the genetic map position. Unit is centimorgan, and y-axis represent low score. And we found the two significant QTL peaks on chromosome 5, 26 centimorgan with a low score of 13.4, and the second largest QTL peaks at 59 centimorgan with a low score of 6.6. And as the Walter mentioned before, I was really happy with getting this picture because I spent around like 5 to 6 years to see this picture. So actually, when I get this one, I complain about taking a lot of time and while discussing with Walter, but even Walter told me, Jay, I have waited for 17 years. So we was really happy with getting this picture. So actually, QTL analysis was completed here. However, we was wondering whether these two QTL peaks represent previously known nematode resistance genes or not. I suspect that this would represent H1 gene because the H1 gene is known to be distal end of the long arm of the chromosome 5. And also I suspect this gene represent, this QTL peak represent GRP1 gene because GRP1 gene is known to be resistant both golden nematode and the pale cyst nematode, as well as it is also located on a chromosome 5. So to characterize these two QTL peaks, I added two additional tests. One of them is PCR test with a 57R marker. So basically, I performed the PCR test for Y36 population with a 57R marker, which is known to be linked to the H1 gene. And then I used the PCR results as a phenotype data for tetraploid SNP map. So I could map the location of the 57R, or I can say H1 gene. Also, thanks to the support of the Dr. Dendron in the University of Idaho, I could achieve the G-polymer resistance data of the same mapping population Y36. So I also load this G-polymer resistance data on the tetraploid SNP map. And this is the result. So the second picture shows the R02 resistance, and third picture shows 57 over H1 gene location, and the last picture shows G-pallida resistance QTL. PA23/2/3 means a mixture of pest type 2 and 3. So when I check the location of H1 gene, as you can see here, it was located on the 66 organ on the chromosome 5, which is very close to the second QTL peak of the R2 resistance. And when I checked the G-pallida resistance QTL peak, it was perfectly matched with the most significant QTL peak of the R2 resistance. So from these views, I also confirmed that this second largest QTL peak represents H1 gene, and this QTL peak represents GRP1 gene. And because, as I mentioned before, GRP1 gene is known to be resistant both golden nematode and pale cyst nematode. And also, I thought that H1 gene appears to provide some resistance against R2, presumably due to the presence of the low percentage of R1 in the R2 population. I just want to mention at this point is that originally R2 obtained from the field, which is initially infested with pest type R1. So the original R2 samples mixed with a small amount of the R1. So I suppose that maybe H1 gene also works for this study. But further analysis, of course, should be following to clarify what happened here. So I also checked the percent of variance explained at each position. When I check the R02 resistance at the 26 centimorgan, I'm going to call for convenience R02A. It shows 24% of variance experience. It's quite high. And when I check the G-polar resistance QTL peak at here, which I'm going to use PA2/3A, and this also shows 9.2% of variance explained. Both of them are quite significant. And when I check this position, R2 resistance peak at here, I'm going to call R2B, and this also explains 13%. So both of them seem to be really important position for R2 resistance. And based on the information provided by the tetraploid SNP, I marked the QTL position on the genetic map. So as I mentioned before, homolog 1 to 4 represent 4 homos of Ni121, and 5 to 8 represent 4 homos of Ni115. And as you can see here, R02A and our PA2/3As are overlap each other. And when I check the R02B and 57 location, both of them are on the same location as well as same homologs, 3 and 6. So I also support the idea that this area includes GRP1 gene, and these two areas include H1 gene. Actually, this is redundant or extra research project, but for double checking these two positions, I also performed the GWAS with the software released by the Jeffrey Enderman's group, RR Blob and GWAS Poly. So basically, I use exactly the same marker data and the phenotype data. And as you can see here, both of them are perfectly matched because the top picture shows a genetic map that I developed before. So the most closest SNP marker was C25031. And the most closest SNP marker to 59 centimorgan was C2454365. And as you can see here, when I check the 57 GWAS, when I check the 57 QTL through the GWAS, the most significant QTL peak located on a SNP marker C25436, which is perfectly matched with this one. And when I check the R02 resistance, there were two peaks, and when I checked their location, this one matched with this SNP marker, and this one matched with this SNP marker. So this GWAS study also support the idea that these two positions are really important for our two resistance. So based on these results, our lab decided to develop diagnostic marker. Actually, Xu Peng, she's a lab technician in our lab, already developed one CAPS marker. Actually, she started from the C25031, and when she tested the newly designed marker with some several resistance clones and susceptible clones, at this point, the results are well matched. So we are going to test more clones, not only in New York breeding clones, but also in other states such as Idaho. We try to apply the newly designed CAPS marker into the cornell breeding program. Okay, now I'm going to start the QTL analysis of chip color and the specific gravity. These two traits are really important in the potato chipping industry because the lighter color potato chip, if you guys open the lace, you can see really white color from the chips. Those colors are really preferred by American. So whenever we do the breeding program, we want to select lighter potato chip color. Also, specific gravity is important since it is always related to starch content and starch density. So this is the reason why I perform this research project with the same population that I used in a nematode study. So I use the same mapping population, Y36 population. The two parents shows quite good color, but especially N115 shows extremely lighter chip color compared to the other cultivar. So this is quite useful clone for selecting or developing chipping potato. And in this case, I used the 195 clones for QTL analysis. Now I'm going to explain how I measure the chip color and the specific gravity. For measuring chip color, I use 9 scale. This is 1, 2, 3, and this is 9, the darkest one. And to measure the specific gravity, I use this equation. Weight in air divided by weight in air minus weight in water. And I have measured during 4 years, from 2010 to 2013. So when after getting the phenotype data, I calculate the Brostein's heritability for each trait. And as you can see here, both of them shows relatively higher heritabilities. Chip color was around like a 7, and specific gravity is 0.75. And also after that, I transformed the original phenotype data to the below. There are a couple of reasons. First of all, the original phenotype data does not show really good normal distribution. So once I transformed to the blob, I could see much better normal distribution like this way. Also, when I performed the QTL analysis with the original phenotype data, I could not see the QTL peaks, which consistently showed up on the same position. But after I transformed to the blob, I could see many QTS which showed up on the same position on the same chromosome. So I suppose that maybe those are because of the G by E effect. So by transforming to the blob, I could more focus on genetic effect. So after getting the blob data, I can compare first of all across the year, and also I can compare. I check the correlation between chip color and the specific gravity. So as you can see in this table, there's no big difference between 2 years. When I see the specific gravity, all correlations around like a 9, so really high. There's no huge difference. And even though chip color shows relatively smaller correlation compared to specific gravity, but still it's quite high. Most of them show over 0.7. So I could not see huge difference across the year. And when I checked the correlation between chip color and the specific gravity, I could see the negative correlation, even though the value itself is relatively smaller. But this is quite good sign for the potato breeder because if a breeder focus on lighter color, that means we can have a chance to select the higher specific gravity, which is preferred by the potato chip company as well at the same time. So I think this is quite good sign for breeding purpose. And then I found a lot of QTL peaks from the many multiple chromosomes. In this case, CC represent chip color and number 2010, 2012 represent the phenotype data from each year. As I mentioned before, I measure phenotype data in 2010, 11, 12, and 13. So these represent each year. And there's all. All means I could get, after I transform to the blob data, I could get 5 different set blob data. 4 represent each year, 2010 to 2013. And last blob data set was obtained by pulling all phenotype across the 4 years. So in this case, all represent the blob data obtained by pulling all phenotype data across the 4 years. So from this table, first of all, I could find significant QTL on chromosome 1, 3, 4, 5, 6, 7, 8, 10, and 12. There are a lot. And interestingly, chromosome 3, 4, 7 shows the QTL peaks, which consistently appeared on same position or very close position. And also, in this case, the solid scale represent the QTL making darker color, and hollow scale represent the QTL making lighter color. Yeah, and this shows where the QTL peaks locate among the 4 homologs. So when I see the chromosome 7, there was largest QTL for chip color, especially homolog 7, which came from the NI115, of course. And this explains 13% of the variance explained for 4 years. So I think this is quite important QTL peak for future chip color selection. And near here, 2 carbohydrate genes, sucrose phosphate synthetase and plasma membrane proton ATPase located within a 1 low support interval for this QTL. When I checked the chromosome 3, there was minor QTL for chip color located at the 67 centimorgan. And a putative invertase inhibitor gene is located about 1 million base pair away from this QTL. This table showed the QTL peak of the specific gravity. SG represents specific gravity. And rest of them are the same. And in this case, arrowhead toward the downside represent lowering specific gravity, and the arrowhead toward the upside represent making the QTL making or increasing the specific gravity. And while analyzing, I realized that chromosome 2, 3, 4, 5, and 8 shows the QTL peak, which appeared on the same position or very close position each other. While analyzing the table, I found that on the chromosome 4, the most significant QTL peaks exist at the year 28 centimorgan, explaining 6.4 to 11% of the variance explained, depending on the year. And a gene encoding phosphofructokinase is about 200,000 base pair away from the QTL peaks. This enzyme play an important role in basically control the first committed step of the glycolysis respiration. So as the starch content represent the amount of the starch synthesis less the amount of the respired away from, this gene become like a really interesting candidate for this QTL. Chromosome 2 has a single QTL at the 72 centimorgan, and a candidate gene, glucose 6-phosphate dehydrogenase, is mapped around like a 500,000 base pair from the target QTL. And chromosome 5 also showed a minor QTL at the 31 centimorgan. A gene coding for starch granule bound RO1 protein is located around 300,000 base pair from the this QTL peak. And silencing this gene is known to increase the starch level in a leaves. So I think this is also quite important for future specific variety selection. So let me summarize this whole presentation. As I mentioned before, these two markers can be used for corneal breeding program. And the QTL at the 26 centimorgan of the chromosome 5 confer races to both RO2 and PA2/3 pest type. And H1 gene appears to provide some resistance against the RO2, presumably due to the presence of a low percentage of RO1 in the RO2 population. And chip color and specific grades were highly heritable, and notable QTL peak for chip color detected on chromosome 3 and 7. Notable QTL peaks for specific grades were observed on chromosome 2, 4, and 5. So that's all for today, and I just want to appreciate my committee member. Thank you for Walter. Long story, make sure that thank you for your patience. And I appreciate Xiaoyang's encouragement. She always encouraged me to start like a youth research project or interesting research project. So we really appreciate it. And Ronnie Kaufman gave me a lot of advice about, advices about international agriculture, because I'm really interested in international agriculture, especially reducing the hunger problem. And he gave me a lot of good suggestions, so appreciate it. And I appreciate the colleagues as well. This is my lab members, and they help me a lot to practice presentation and to do the research project. And technicians are amazing because these guys works really well and support me a lot. So I appreciate Shuping and Matt. And also Michael Muzeric gave me a chance to do the TA at first a long time ago. But yeah, so I appreciate because of his he gave me the chance. I could practice, especially like I could get the teaching experience. And Darlene helped me to practice this presentation, so I appreciate it. And Denise helped me a lot for statistical analysis. So I learned a lot of important statistical methods from him. And nematologists that support me a lot as well, since I did not have any experience in a nematology, but Xian and Fusion support me to do the experiments, so I learned a lot from them. And also Michigan group support me a lot for genotyping and do the QTL analysis. And also Luis helped me to get the G-Polydor data, so without her support, I could not complete this research project. And Chris Neckett, she's a software developer. She developed the tetraploid, and she support a lot of American researchers. Actually, she stayed in Scotland, but she frequently visit the US and then communicate with many American researchers. And she also helped me a lot. And also Jeffrey Endelman helped me to do the GWAS analysis. So finally, I appreciate whole Cornell community because, you know, I was not like a super smart student when I came to here first, but the Cornell community changed me a lot. Whenever I go to the conference, I have a really, how can I say that? Feels like I'm really competitive. I know it's quite a funny story, but you know, Cornell community is amazing. I can guarantee that. So I appreciate it. Thank you for listening to my presentation. Thank you.
Any questions for Jay?
No question.
I'd like to ask a question. That is, you may have covered this in many tracks, but one of the things which is a reservation I have in the use of the markers to pick out H1 resistance is that the Ernie was the primary gene
Yes.
species that was used in the breeding program before I came. And it was the major effort was on Ernie resistance before the H1 gene was identified and the antigen. So there's there's a lot of clones that are, when we were using the bio test for resistance, we picked out resistance, and we did not know whether it was Berniae or
Yes, we cannot know.
I think just through time and simplicity, H1 predominated still. But I'm afraid that when we throw away on the marker, those that do not have H1 that are resistant, we're losing something. And without doing a bio test on the susceptible clones for H1, the absence of H1, we won't pick them out and then wait. And now maybe I didn't realize it was all the I was resistant when we use it in insect. But it doesn't bother me. I'm happy that it's there.
Yeah. So yeah, you're right. H1 gene originates from the Solanum indigena, especially CPC 1673 something. So according to my knowledge, at least, almost all breeding programs use that specific clone, Solanum indigena CPC 1673. So I think the H1 gene that we had also came from indigena, and as you told me, that S. Berniae, there's another wild potato. That is also known to have H1 gene. But again, the case 4. Yeah, the case 4. Yeah, I cannot clearly explain what happened in these clones, but Walter and I were discussing. We suppose that maybe, because as I mentioned before, S fertility also has an R1 according to the one European report. So maybe another resistance is located in another chromosome affect the R1 resistance. Yeah, but I don't know. Yeah, what happened here.
You said the Eva does not have the H1 gene. What you said?
Eva has actually. I believe that Eva has a H1 gene, but interestingly, this was negative for TG689.
Jaebum can correct me if I'm wrong. I think the plan is that she will test those that all. Mark will be the first screener. Jaebum will test the susceptibles to make sure.
That's our current practice. We need to use marker to provide screen, and then basically for those showing negative for both markers, we will do that just to be sure that we don't miss anything that are not resistant.
So you talked about like how much variance you could explain with the specific QTLs you identified. If you try using all the markers like the regression model and saying that how much is explained when you use the whole marker set?
No, generally like that way. Of course, technically we can calculate for each marker, but it takes a lot of time, frankly speaking. So the software, the tetraploid map software always provide QTL. Actually, he asked about did I calculate percent of variance explained for each marker? Am I right? Whole genome. Yeah. So I can say I want to say that I didn't, but I don't need to do. Let me explain it. So in the tetraploid software always provide the most significant QTL picks for each chromosome. So once I put the appropriate genotype data and then phenotype data, this number and where it is located are automatically showed up on the screen. However, if you really want to calculate other position, for example, if you want to know how much this area explain percent of variance explained, you have to make a kind of like a linear model for taking out some clones which has alleles related to this one. Because if you keep all population to calculate this one, the software does not show percent of variance explained for this area. Always shows the most significant QTL peaks only. So I can say that. Really, I have to explain all the long story. I mean, you have to develop the linear model and then figure out which clones are related to this one, which clone is not related to this one. So once you found the clones related to this one, you have to take it out from the population genetic mapping, and then you have to rerun the whole QTL analysis. So really complicating. Also, 24 is extremely high whenever I do the QTL analysis for other traits. So I guarantee that other area has a really small percent of variance. It's almost ignorable.
Yeah, so that's why I use the genome selection model. You can predict that you can't identify which areas are associated, but just predicting.
Yeah.
You can use like hard luck just to predict.
Just predict for another area, you mean?
Just like predicting the phenotype of all your markers.
Or your yeah, it's possible. It's possible. Yeah. So you mean maybe the genotype data also use as like a kind of like a phenotype data, you mean?
Yeah.
Okay. So yeah, so your comments is really useful as well as it's possible, but I just want to emphasize that I work in a potato breeding, especially we have a specific breeding purpose. So we are only interested in the position which is most related to the target trait. So even though maybe other area would be related, would explain small amount of variance, but for the breeding purpose, those are ignorable. So we just focus on the two significant QTL. Thank you again.
This has been a production of Cornell University, on the web at cornell.edu.